们的活动即将开始，请入座。活动开始前，请各位先将手机调为震动，感谢您的合作。Ladies and gentlemen, our event will begin shortly. May I invite you to take your seat? Before we begin, please switch your mobile phones to silent mode. Thank you for your cooperation. Ladies and gentlemen, distinguished guests, and dear professors and students from NTHU, good afternoon. Welcome to today's keynote speech, delivered by Professor Brian Kobilka, 2012 Nobel Laureate in Chemistry. This lecture is part of the TAIWAN BRIDGES event series. Welcome to today's keynote speech, delivered by Professor Brian Kobilka, 2012 Nobel Laureate in Chemistry. This lecture is part of the TAIWAN BRIDGES event series. We hope this event will inspire your thinking and emphasize the importance of continued support for basic science. It's our great honor to have you all here today. We would like to give a special thanks to Academia Sinica, TSMC, and various university and corporate partners for their joint sponsorship. To officially begin today's event, we are honored to have with us President Chung of National Tsing Hua University. Ladies and gentlemen, please join me in welcoming President Gao to deliver his remarks.
Good afternoon, ladies and gentlemen. Welcome to NTHU. This is the 20th year of our series on Nobel Laureate and NTHU, and we are delighted to have with us over the years almost 50 different Nobel laureates from all disciplines, sharing with us their stories, their struggles, their triumph, and their dreams. And today we are especially happy to welcome Brian, Dr. Kobilka from Stanford University, to share his story with us. And we're especially delighted to have the Bridge program as our co-sponsor for this very important event. And I guess one thing that always dawned upon me is what it takes to be a Nobel laureate. I think first and foremost is to have a kind of a free environment for multidisciplinary education and research. That open-mindedness is absolutely critical. Yesterday, we heard a wonderful talk from Dr. Prof telling us about his interdisciplinary research on kidney transplant. But he's an economist, and his award was in the economic sciences. So today, I'm sure Brian will share with us his journey in drug delivery and drug development, and that will involve many, many different disciplines coming together. So with that, we welcome Brian to NTHU, and I look forward to a very successful event. Thank you.
Thank you, President Gao. To express our warm welcome and sincere appreciation, we have prepared a commemorative gift for Professor Kobilka. Please join me in warmly welcome Professor Brian Kobilka. We now invite President Gao to present the gift, a tie featuring the Chinese character Ching, symbolizing National Tsing Hua University, as a token of our deep appreciation. We would also like to invite President Gao and Professor Kobilka to take a photo together to capture this special moment. Thank you. May we invite President Gao and Professor Kobilka to return to their seat. We would now like to invite all distinguished guests, faculty members, and students to remain seated and join Professor Kobilka for a group photograph. Next, please join me in welcoming our host, Senior Vice President of NTHU, Professor Lü, who will introduce our speaker.
Friend and all the distinguished guests, welcome to Tsing Hua. It's a profound honor to introduce Professor Brian Kobilka, a scientist whose remarkable journey began in a family bakery in Minnesota, and led to the pinnacle of scientific achievement. Professor Kobilka earned his bachelor's degree in chemistry from the University of Minnesota, Dulles, and his MD from Yale University School of Medicine. Following his residency in internal medicine, he joined the laboratory of Robert Lefkowitz at Duke University, where he began the transformative research that would eventually change the world of medicine. Currently, he serves as the professor of molecular and cellular physiology at the Stanford University School of Medicine. In 2012, Professor Kobilka was awarded the Nobel Prize in Chemistry for his pioneering work on the structure and mechanism of G-protein coupled receptors, GPCR. His research provided the first atomic level insights into how drugs interact with this vital signaling system, providing a blueprint that has fundamentally revolutionized modern drug design. Today, the impact of his work resonates across cardiovascular medicine, psychiatry, and neuroscience, bridging the gap between basic science and the clinical breakthrough. Please join me in a warm round of applause as we welcome Professor Brian Kobilka to deliver his talk.
Thank you very much for inviting me here to talk to you today. I want to really talk a little bit about, in the beginning, a brief overview of what GPCRs are. And I know that the audience is a mixed audience. And so I want to give you a little background on the history of G-protein coupled receptors and an overview of what these receptors are and why they're important. And then in the second half, I'll talk about the application of structural biology. The application of structural biology and how it may impact drug discovery. And I'm using the mu opiate receptor as my model system. So what are G-protein coupled receptors? So they're proteins that reside in the plasma membrane, and they transmit signals across the cell membrane to change the behavior of cells. So this was a cartoon of what we knew about the field when I joined the Lefkowitz lab in 1984. We knew there were proteins in the membrane, plasma membrane, that when activated by a hormone or neurotransmitter would engage a heterotrimeric G protein. So those are G proteins consisting of alpha, beta, and a gamma subunit. And in doing so, they would activate the G protein, and the G protein would release GDP, and bind GTP, and that would then cause the G protein to break apart into two components, an alpha subunit and a beta gamma subunit. And both of these can go on to modify the behavior of a number of proteins in the cell, particularly enzymes and ion channels. And this activity continues until the GTP is hydrolyzed to GDP, and then the cycle can begin again. And this is somewhat of an oversimplification, but it probably applies to almost all G-protein coupled receptors. So G-protein coupled receptors mediate the majority of cellular responses to hormones and neurotransmitters. So briefly, what are neurotransmitters? So they're small molecules at the end of neurons that allow one neuron to talk to another neuron. And some of these neurotransmitters include things that you've heard of, including adrenaline and noradrenaline, dopamine, serotonin, glutamate, endogenous opiates, and endogenous cannabinoids. Hormones are, again, small molecules typically, or small peptides, released from one cell and can change the behavior of cells in the local environment, or can change the behavior of cells at a distance by going into the bloodstream and circulating throughout the body. And some of these local signalers include histamine and adenosine and prostaglandins, and more distant include adrenaline, angiotensin, glucagon, and various gonadotropins. The system that I became most interested in as a physician working in the intensive care unit is that we were using drugs that worked on a specialized system in the body called the autonomic nervous system. So the autonomic nervous system was designed to help your brain control a variety of functions, including your heart rate, contractility, airways, gut, intestines, and this system consists of activating neurotransmitters and inhibiting neurotransmitters, more or less. So the sympathetic system is what we call the fight or flight system. Activation of the system leads to an increase in heart rate, increase in blood pressure, shunting of blood from the kidneys and intestines so that all the blood goes to the skeletal muscle and prepare you for fight or flight. So there are 9 adrenergic receptor subtypes, and the receptor that I have worked on earliest in my career, the beta-2 adrenergic receptor, is a receptor that is found in rich supply in lungs, and drugs that activate this receptor are used to treat asthma. The parasympathetic system has sort of a counteracting system. It's often called the rest and digest. It decreases the heart rate, decreases blood pressure, and increases blood flow to kidneys and intestines. And if you work in an intensive care unit, you're often administering drugs that either activate this pathway or what are a member of this pathway. And there are 5 muscarinic receptor subtypes. So for example, in the heart, blowing this up, the heart has special sensors, both in the atria and in the aorta, that measure blood pressure. And this information, let's say for some reason you've lost your blood pressure, it drops, you've had an accident, you've been bleeding, that information is then sent to the midbrain, and the midbrain sends information to the heart to increase the contractility, increase heart rate, and so essentially sympathetic nerve terminals at cardiac myocytes. So in addition to their role in mediating cellular responses to hormones and neurotransmitters, they mediate the senses of sight, smell, and taste. And there are over 800 members of the G protein coupled receptor superfamily. So I find this particularly interesting. So that these are the 800 members of the human GPCR. Of all of these 800, the majority of these receptors, the largest number of receptors, are dedicated to the sense of smell, olfaction. So humans don't really, sense of smell is important, but we really don't need it to survive. However, if you're an animal that does need it to survive, either to find food or avoid being food, you have even more olfactory receptors. So for example, the mouse and the rat have roughly 1,000 olfactory receptors. So what surprised me when I found out the animal that has the most olfactory receptors, it might make sense, it's the animal with the biggest nose. So elephants have roughly 2,000 olfactory receptors. And I'm not really sure why that is, but they have very poor visual acuity, and it might help with their, essentially they're staying together as a herd. So you can see there's different color codes. These reflect different subfamilies. The largest subfamily that includes the olfactory receptors also includes receptors that we're going to talk about today, including adrenergic receptors and opiate receptors, particularly the mu opiate receptor. But it also includes other members, including receptors for dopamine, serotonin, cannabinoids. And there's another family of receptors, subfamily called family B, that include receptors that you're probably really familiar with. At least you're familiar with the drugs that activate these. These are GIP and GLP receptors that are used to treat diabetes and obesity. So because they're such a large family, they're so important in regulating so many functions in the body. They're the single largest class of drug targets. And I'm going to talk more about that towards the end of the talk. But roughly 30 or 35% of all drugs in the market target G protein coupled receptors. And it probably would be more if we were more successful at developing drugs for these receptors. So a brief history of the field. This is more or less from my perspective. But I think the field really began in 1971 when Martin Rodbell discovered that guanine nucleotides were necessary for the signaling of certain hormone receptors. It wasn't until 1977 when Ross and Gilman biochemically isolated the first G protein. And Rodbell and Gilman would ultimately win the Nobel Prize for their work on G proteins. In 1984, the Lefkowitz lab purified the beta-2 adrenergic receptor from hamster lung. And it was a very fortunate time for me. I joined the Lefkowitz lab when this really important discovery was made. That discovery by a graduate student in the Lefkowitz lab, Jeff Benovic, allowed us in 1986 to clone the beta-2 adrenergic receptor. And that gave us the first clue as to what this protein looked like. We knew that it had 7 transmembrane domains, and we actually, for the first time, realized that it looks a lot like rhodopsin, the visual pigment in the eye. So after spending 5 years in the Lefkowitz lab, in 1990, I started my own research program at Stanford. And my goal at that point was to try to determine the structures of receptors and receptor G protein complexes. The first structure that was obtained of the signaling pathway was in 1995, and it was a structure from the Gilman lab on the heterotrimeric G protein. But it wasn't until 2007 that we had our first success in obtaining a structure of the beta-2 receptor in the inactive state. So this was, for me, starting in 1990 to finally having success in 2007, a very long, protracted journey to accomplish this goal. And as you'll see in a few slides, I wasn't alone. I had a lot of great collaborators in this effort. In 2011, we determined the first structure of an active state GPCR, and then finally in 2011, the structure of the beta-2 G protein complex. So these are the critical structures that we obtained, that were obtained and that allowed us to, for the first time, really assemble the process of receptor activation. So the inactive state, active state, and receptor G protein complex, they all have 7 transmembranes, and this is a signature that all G protein coupled receptors have. So as I mentioned, this is a very long process, and it involved collaborations with a number of individuals throughout the United States and Europe, and even in Korea. So first of all, I was fortunate to have expertise in G protein biochemistry. My lab would work very hard on obtaining sufficient amounts of high quality beta-2 adrenergic receptor, the receptor itself. And at a Gordon conference, Roger Sunahara approached me and said, you know, we should really start working together. And so this was a really fortunate collaboration. We also identified a very high affinity agonist by testing over 60 compounds screened from a number of pharmaceutical companies. So when you want to purify a membrane protein like receptors, you have to extract them from the membrane. And you do that by using special detergents. And those detergents are incredibly important. Most of the detergents that people might use in their biochemistry labs will kill a G protein coupled receptor. And we were very fortunate to have this detergent called MNG3, or LMNG as people might know it now. It was obtained from Hilsup Che, who's a detergent chemist. So nobody else had access to this particular detergent. It was part of the collaboration. We also used small antibodies from llamas called nanobodies to stabilize the complex. And the project was largely guided by negative stain EM. Back then we didn't have cryo-EM, but we could use negative stain EM to make sure the protein looked uniform. And then finally, we used a special kind of crystallization method called lipidocubic phase, and a special lipid, again, that we were able to get, not commercially, but through a collaboration with Martin Caffrey. So there are even more individuals that are involved as well, including Bill Weiss, who has always helped us with crystallography. So what did we learn from the beta-2 receptor about the activation process? So first of all, I'm simply rotating the receptor. The agonist adrenaline is shown on top in spheres. The G protein, alpha is orange, beta is in cyan, and gamma is in purple. And I'm going to toggle back and forth between inactive and active states just to point out a couple things. So when the receptor is bound to an agonist, there's quite a small change. So the agonist binding pocket is formed by the extracellular side of these transmembranes. And when the agonist binds, there's a small contraction of the binding pocket, about 2 angstroms. But that leads to the propagation of a much larger change, and particularly in transmembrane 6 in the cytoplasmic domain. And when that transmembrane 6 opens up, it allows the engagement of the very C-terminal alpha helix of the G protein. And this leads to activation, propagation of conformational change and activation of the G protein. So the G protein itself consists of 2 domains, a RAS domain and the alpha helical domain. And you'll see this incredibly large conformational change upon activation. The guanine nucleotide resides between the RAS domain and the alpha helical domain. And when that dissociates, and this is what we call a nucleotide-free state, when that dissociates, the alpha helical domain becomes highly dynamic. And this is one of the reasons why it was so difficult to crystallize this receptor, because having a component that's that dynamic will really often prevent you from forming crystals. So at this time we had a receptor, a specific receptor, the beta-2 receptor, coupled to a very specific G protein. And I mentioned there are 800 receptors. There are quite a few of G proteins as well. So there's the alpha S family, and this was the G protein that we crystallized with the beta-2 receptor. There's the alpha I family and alpha Q and alpha 1211 family. And so we wanted to know if what we observed for the beta-2 receptor was also observed for other receptor G protein complexes. So we looked at other receptors, muscarinic receptors that couple to the GI and the GO family, opiate receptors that couple to the GI family, and I don't have it here, but we also have a receptor that couples to the GQ family. And we were able to make really beautiful complexes, biochemically stable and pure. However, we were not able to grow crystals. The only receptor G protein complex that we've ever been able to grow crystals of was of the beta-2 GS complex. So why is that? Well, in order to determine structures by crystallography, you have to have your purified protein. You have to coax it to forming crystals. And this is the major challenge. Your protein has to be pretty rigid. It can't have a lot of flexibility. But if you are successful, you can then take it to a synchrotron, shine X-rays through it, and it will produce a diffraction pattern that can be then used to determine the structure of your protein. But as I mentioned, there's probably one domain of all of these receptor G protein complexes that's highly dynamic, that's this alpha helical domain, and it was preventing us from getting crystal structures. However, around 2017, we were able to collaborate with Jirvis Kiniotis with obtaining structures by cryo-EM. because of advances in cryo-electron microscopy, including direct electron detectors, so that you don't need to form crystals. You just simply have a high concentration of your protein in solution, and you diffract electrons, and that's detected in, again, this direct electron detector. You have methods to correct for motion as a result of heating of the sample. And then there are a variety of computational methods that help you take these individual molecules and convert them into a three-dimensional structure. And after having access to cryo-electron microscopy, we were able to get a large variety of receptor G-protein complexes, cannabinoid receptor, opiate receptor, muscarinic receptors, as well as members of other family, including family B and family C. So what did we learn? And I should mention that we now have, I think, well over 100 structures, not from my lab, but for other labs throughout the world, because it's become relatively easy to obtain receptor G-protein complex structures. So what did we learn? So first of all of these structures are somewhat artificial in that they are of a nucleotide-free state of the receptor. So in a cell, when the G-protein releases GDP, there's a high concentration of GTP in the cell, and it's rarely nucleotide-free for very long. But unless it's nucleotide-free, it's not stable. So if you have either GDP around, it will dissociate in this direction. If you have TP around, it will dissociate in this direction. So there are some, I would say, artificial features of our receptor G-protein complexes, but for the most part, they're quite accurate. Structural changes in the different G-protein isoforms are the same or very similar. The alpha helical domain of the G-proteins, all the different G-proteins subtypes shown here, is flexible and dynamic, and again, probably why we couldn't grow crystals. What's a bit disturbing is that we have all these structures. Unfortunately, the GPCR, G-protein complex specificity, can't be fully explained by these structures. So why does the beta-2 receptor prefer GS or GI? Why do muscarinic receptors prefer GI over GQ or GS? And we think that the reason we're not seeing evidence for specificity is that we're missing structures. So these two structures represent what we think are intermediate states, and we now have some biochemical and biophysical evidence that, in fact, there are at least two intermediate states that probably determine which G-protein a receptor is going to couple to. And we're currently trying to obtain those structures. So this is my background for a brief overview of the history of the GPCR field. I'd now like to talk a bit about the challenges in drug discovery. So particularly applications of structure information and structural biology to facilitate drug discovery. But before I do, I want to talk about the challenges that we face in drug discovery for GPCRs. So first of all, signaling pathways can be quite complex. And while the beta-2 receptor has a relatively simple signaling pathway, one receptor coupled to the G-protein GS, the mu-opiate receptor is quite complex. So the mu-opiate receptor can couple to 6 different G-proteins, and it can couple to 2 different arrestins. So G-proteins are the major signaling pathway. Arrestins are the major pathway by which receptors are desensitized. So often activation of the G-protein is first, then you get phosphorylation of the receptor by a particular family of kinases. That leads to engagement of the receptor with a protein called arrestin, internalization of the receptor, and either degradation or recycling back to the plasma membrane. It's also possible that in some cases, arrestin itself is also a signaling molecule. So while it's internalized, the receptor may continue to signal through a slightly different pathway than through G-proteins. So the question here is, you have all these G-proteins and arrestins, which are responsible for the therapeutic effect, and there's really only one therapeutic effect, and that's analgesia. And which are responsible for the adverse effects? Respiratory depression, sedation. So the reason people die if they take an overdose of opiates is they simply stop breathing. Sedation, addiction, tolerance, and constipation. So what I'm going to talk about a little later on is our efforts to try to understand the signaling and which pathways are therapeutic. So the other problem is complex pharmacology. We don't simply have agonists and inhibitors. We have a full spectrum of compounds that can regulate the activity of a receptor. So for example, if this was the opiate receptor, first of all, we start with some level of activity, even in the absence of an agonist. So this is called basal activity. And most GPCRs have some level of basal activity. There are drugs, such as naloxone, that can suppress this basal activity. There are also drugs that are neutral antagonists. They don't activate or they don't inhibit. But most of the therapeutic drugs are either full agonists or strong partial agonists. And these include fentanyl, which I'm sure you've heard about. Morphine and heroin, which are quite effective at relieving pain, are actually partial agonists. And buprenorphine, which is now used to treat opiate addiction, is a very weak partial agonist. But it turns out to be the highest potency. So other things that we have to worry about are pharmacodynamics, efficacy, affinity, dissociation, half-life, and pharmacokinetics. So how long first of all, is the drug orally available, its rate of absorption, elimination, and blood-brain barrier penetration. So all of these have to be, have the right activity to have a drug be successful. Cell and tissue-specific signaling. So a drug like, a receptor like the beta-1 and beta-2 receptors both couple to GS, but in the cardiac myocyte, they don't have the same signaling pathways because it's not because of the G protein that they couple to, but it's more that where they reside in the membrane. So there are some things called signaling domains. And the signaling domain, for example, for the beta-2 receptor in the heart may be different in the lung or in the blood vessels. And this can also change the behavior of your therapeutic. High degree of sequence similarity in the orthosteric pocket. So the orthosteric pocket is the binding pocket for the native hormone or neurotransmitter. And this is one of the biggest challenges in finding drugs that are selected for your target. So this is another version of the family tree, just of the family A members. And we're going to focus on this quadrant and blow that up. And what I highlight here are adrenergic receptors shown in red, muscarinic receptors shown in cyan, serotonin in blue, and green dopamine. And what you'll see is a cluster of receptors here that are highly homologous. So the adrenergic receptors are closely related to the dopamine receptors and the serotonin receptors. And if you develop a drug, say, for a serotonin receptor, it's very likely to have some off-target effects on the adrenergic or a dopamine receptor. And then finally, as more and more genomes get sequenced, we realize that we don't all have the same opiate receptors. Some of us have slight modifications that can affect the way we respond to drugs. So this is a single point mutation that's found in a number of individuals that greatly reduces their response to drugs. So this is endomorphin, an endogenous opiate, but this is morphine. You can see morphine is quite ineffective, as is buprenorphine, because of this mutation that's at the bottom of the binding pocket. So we became interested in the opiate receptor for a number of reasons, but particularly because it is becoming a major problem, particularly in the United States, with deaths from opiate overdose escalating, particularly with the availability of fentanyl. So again, our goal was to try to understand the various signaling pathways that the receptor can engage, and to ask which signaling pathways can we target for beneficial effects, and which can we try to eliminate because of the adverse effects. So I'm going to first talk a bit about the structure of the opiate receptor itself. It's somewhat unusual, and why it's a very promiscuous receptor. So this is a surface rendering of the opiate receptor, highlighting the binding pocket shown here. And many of the studies that I'm going to talk about from now on were done in collaboration with Susurta Majumdar and Jay McLaughlin. Susurta is an amazing medicinal chemist, and he's made a number of compounds that I'll show you today. So because this binding pocket is so large, there's a wide spectrum of agonists for the receptor. It includes metenkephalin, which is an endogenous opiate agonist peptide. So the opiate receptor is actually a peptide receptor, but most of the drugs we give to the, we administer that activate opiate receptors are small molecules. So these are some alkaloids from the opiate poppy, including morphine and buprenorphine. There is a non-opiate alkaloid called metragenine pseudoindoxyl from the kratom leaf. This actually turns out to be a quite effective pain reliever with relatively few adverse effects. Then there are completely synthetic compounds, including methadone, fentanyl, and the most potent and lethal drug is lofentanyl. And it's only used to treat very, very large animals like elephants or rhinoceros. So we first want to understand why this drug, it's quite effective at relieving pain, but why is it relatively safe, and why is this drug have such severe adverse effects? So as I mentioned, we studied different G proteins they activate, and these two receptors have very different activation pathways. So this is simply a dose response of the agonist lofentanyl or metragenine to the various G protein subtypes and for arrestins. And you can see lofentanyl is basically a super agonist for activating all 6 G proteins and 2 arrestins. In contrast, for metragenine pseudoindoxyl, it's only a partial agonist at all of the G protein subtypes, but of interest, it doesn't activate or engage arrestin at all. So it's what we call a G protein biased receptor, agonist. So we wanted to know if we could learn something about how they engage the receptor binding pocket and understand why they're so different in terms of their behavior. So this is, these are 2 structures we obtained by cryo-EM, looking at the outside, from the outside of the cell, looking at the binding pocket, and you can see again it's a very open binding pocket, and both lofentanyl and metragenine occupy pretty similar space, but there is one space that's occupied by metragenine, but not by lofentanyl. So if we zoom in on the binding pocket, you can see that in lofentanyl, as a result of where the lofentanyl binds, and that's shown in blue here, you form this very strong polar network, hydrogen bonding network, from transmembrane 2 through transmembrane 7. In contrast, because of the shape of metragenine, it inserts into this space, this other space that I mentioned on the outside, and that basically prevents the formation of this hydrogen bonding network. And as a result of the hydrogen bonding network, you can see there's a slight rotation in this tyrosine in transmembrane 7 that doesn't occur in transmembrane 7 of metragenine occupied receptor. And as a result of that, in studies that I won't be able to show, we actually looked at on the inside of the cell and tried to understand structural changes in the part of the receptor that engages G protein. And what we found is that, as expected, there was a large outward movement of TM6. If you remember when I showed you the activation of the receptor, TM6 undergoes the biggest structural change in the beta-2 receptor and in the mu opiate receptor bound to lofentanyl. Surprisingly, metragenine doesn't cause any outward movement of TM6 at all, nor does morphine and a number of other agonists. And we still don't fully understand, without that key mechanism of activation, how does metragenine activate the receptor. And we're beginning to understand that there are other domains on the inside that, that undergo conformational change as a result of binding to metragenine. And we think those are probably explaining differences in G protein coupling selectivity. So now that we know how drugs bind, Siswara Majumdar wanted to see if he could take, say, a relatively safe drug and make it even better, and using the structure to help guide optimization. So his idea was to modify existing agonists. So this is the binding pocket for mitragynine. You can see there's space below and there's space above, and he's generated agonists that extend into this pocket, as well as agonists that extend in this space as well. And I'm going to talk about one of these we call SC103. And it looks like it's sort of just suspended in space. And if we get a structure of the receptor, we actually see this component, but we don't see this component because it's probably very flexible. Nevertheless, it has a pretty dramatic effect on the behavior of this compound. So below, again, are the dose responses for mitragynine for activation of the various G protein subtypes, as well as arrestin 1 and arrestin 2. And what was surprising is that this would seem like a relatively simple addition of an aromatic here resulted in a pretty dramatic effect on signaling. So now, receptor activated by mitragynine, or SC103, we see that all of the activation of all three G proteins, GZ, GO, and GB, are partial. And it doesn't activate GI1, 2, or 3, or beta-arrestin 1 or arrestin 2. So now we have somewhat of a selective, pathway-selective drug that can only activate 3 of the 6 G proteins. And surprisingly, even though it's a relatively weak partial agonist, this drug can achieve the same level of analgesia as morphine does. So it's a quite effective analgesic, and it does not cause respiratory depression. I'll describe this in a bit later on. But so even though it's a weak partial agonist for only 3 of the 6 G proteins, it's still capable of achieving near maximal levels of analgesia, suggesting that you don't need to fully activate specific G proteins to get analgesia and pain relief. So with these studies that I just mentioned, as well as a number of other studies from Siswara Majumdar's lab, we can begin to guess which G protein pathways are beneficial and which produce adverse effects. And these include analgesia. We know GOA and possibly GZ are essential for activation of the analgesic pathway. We also know that partial agonists can be very effective at analgesics, but produce fewer side effects. Respiratory depression and addiction, we know less about what's causing this, but we suspect it's GI1, 2, or 3, with also needing GZ. And finally, tolerance. The fact that giving multiple doses of an opiate agonist will reduce the relief of pain and require higher doses. Tolerance is caused by this arrestin interaction. So I'd now like to talk a bit about what you would think about how can structures help with drug discovery. And that's in silico screening. And we underwent a project with Brian Shirket's lab a number of years ago. But first of all, what is conventional drug discovery? So compound library sizes are roughly 1 million compound. It has limited scaffold diversity, and it's necessary to experimentally test each compound on cells expressing the receptor. And the whole process is slow and expensive. In contrast, structure-based drug discovery, particularly in silico screening, use docking algorithms together with crystal and cryo-EM structures. The compound library size is now approaching 1 billion. There's high scaffold diversity, and the whole process is faster and less expensive than conventional screening methods. So we simply have a virtual compound library. We test each compound in literally thousands of different orientations, and if you get a hit, you call it a hit, and if it doesn't fit, it has a low score, you reject it. So this was the project that we did with French Ricketts Lab. This was done a number of years ago. At the time, the library size was 5 million, so now it's much larger. The docking was done at the Ricketts Lab. He selected the top 50 compounds based on their scoring function. And then binding and signaling assays were done in my lab. Chemical optimization was done in the lab of Peter Gemeiner in Germany, and then went on to animal studies. So this shows the binding pocket of the mu-opiate receptor with all of the top scoring hits. So they all bound in slightly different ways. And what we did was take all of these and simply look for things that actually had function. In other words, there was evidence that they bound to the opiate receptor. And we highlighted compound 7. So compound 7, shown here, it's a 2 micromolar affinity. Now, the larger that number, the poorer the affinity. So we want to actually reduce this number, and we did an analog screen and found a compound that is racemic and had this modification. And this now was about 10-fold more potent. We did stereoselectivity, and finally, adding a hydroxyl, we got down to 3.7 nanomolar, so almost a 500-fold improvement in potency. And one of our goals in this screening was to find G protein biased compounds. So we weren't looking for particular G protein subtypes, but we were looking for compounds that activated the G protein but not activated arrestin. So here's our PZM21 is the name of our compound, and here's PC21 effect on G protein. So damgo is a peptide agonist. That's sort of the reference point for a full agonist. Morphine, shown in red, is almost a full agonist, but it's a strong partial agonist. And PCM21 is slightly less efficacious, but more potent than morphine. What we were happy to find is that PCM21 did not engage, did not cause the engagement of arrestin. So here again is arrestin recruitment. Damgo is a full agonist. Morphine is a weak partial agonist, and PCM21 had no effect at all. So then, how does this work in animals? So this is a hot plate assay. Simply you're putting a mouse on a plate that's 50 degrees, and you look at how often it licks its paws. And morphine causes quite a strong analgesic effect, and PCM21 is almost as effective, and it has a longer lifetime, which you might expect because of its effect, its lack of effect on arrestin. But perhaps most importantly is respiratory depression. So this is the breathing rate. Animals are injected, and initially they have a very fast breathing rate because they've been handled. But you see if they get morphine, the breathing rate drops dramatically. However, there's no such effect of PCM21, or it's basically the same as vehicle control. So this is quite a desirable effect, and we also know that it doesn't cause addiction to this particular drug. So finally, at the end, I'd like to just mention a new area, a new avenue for drug discovery in my lab, and that's identifying allosteric modulators for the mu-opiate receptor. So what are allosteric modulators? So shown here is a surface view of the mu-opiate receptor. The orange structure here is metenkephalin. It's what we call the orthosteric binding pocket. And blue are residues that form the orthosteric pocket. Allosteric modulators can bind any place else, and simply by binding elsewhere, they might have some effect on the receptor activity. So allosteric modulators bind outside of the orthosteric binding pocket. Allosteric modulators can augment. We call them positive allosteric modulators, or inhibit negative allosteric modulators, physiologic responses to natural hormones and neurotransmitters. They minimize the effect of tonic receptor activation. So if you take an allosteric modulator by itself, it won't activate the receptor, but it will enhance the effect of your endogenous opiates. There's a potential for greater subtype selectivity. Most opiate receptor family members are very similar in the binding pocket, but quite different outside of the binding pocket. And it turns out there's a special kind of library screen called DNA-encoded chemical libraries. They're ideally suited for the identification of allosteric modulators. So what do these modulators look like and how do they work? Well, the red trace here is simply a dose response curve to an orthosteric agonist, and that could be methamphetamine. So you see, as you increase the concentration, you get a higher response. Allosteric modulators can do a couple things. They can either increase the efficacy without changing the potency, or they can improve the potency without changing the efficacy, or they can do both. So what we're hoping to do is find allosteric modulators that modify the effect of endogenous agonists. And one advantage other than the fact that they simply enhance and augment natural hormone synthesis or responses is that we hope that they won't ideally affect signaling through like fentanyl or morphine. So how did we identify drugs? So we became interested in using DNA-encoded chemical libraries a number of years ago. So these libraries are very large libraries of compounds that are generated by adding a fragment to a piece of DNA that tells you what that fragment is. You then pool those together, you add additional fragments. You do this several rounds until you get 3 or 4 fragments bound to a piece of DNA that encodes exactly what that chemical structure is. So the libraries basically covalently attach small molecules to the end of DNA strands containing sequences that code for those molecules. The larger the number of cycles, so this is a 3-cycle compound, the larger the number of cycles, the larger the library size, but also large, for example, 4-cycle compounds are less drug-like. They're often too big and hydrophobic. Okay, so how did we screen? We obtained a library from a company called Wuxi. Again, I'm sure you're all familiar with Wuxi. And it had 4 billion compounds. And so we did a screen where we did it with completely purified protein. So receptor, the mu opiate receptor coupled to GI protein was mixed with the library. Things that bound, things that didn't bind, we washed away. Things that bound, we eluted by heat inactivating. We then took the eluted samples and repeated this 3 or 4 cycles until at the end we had greatly enriched compounds that actually engaged the receptor. And we sent these to Wuxi, and they did the sequence analysis, and they gave us the results. So we didn't just do this with one receptor. We had targets and anti-targets. So our target for PAMs was the mu opiate receptor coupled to the G protein GI. Our anti-target for PAMs was the mu opiate receptor bound to an antagonist. And then we needed an anti-target for the G protein. And this was the cannabinoid receptor coupled to the same G protein. And if we were looking for NAMs, this was our target and this was our anti-target. So we screened this 4 billion compound library. We selected 5 compounds, and we got 2 hits, which is a pretty high hit rate. The hit rate, for example, on an in silico screen might be 0.1%. And you might have to test thousands of compounds. Here, we simply picked, we selected 5 compounds. So the first one I'll show you is a negative allosteric modulator. And it's binding right on top of the orthosteric pocket. So this is a potential interest when a person who's overdosed comes into the emergency room, they get naloxone, and naloxone will displace fentanyl, and the person will wake up right away, often go through withdrawal, and often leave the emergency room. But naloxone doesn't hang around very long, and fentanyl does. So patients often, again, succumb to a fentanyl overdose simply because naloxone is eliminated. But this compound looks like it might actually enhance the stability of naloxone in the binding pocket. And that's exactly what we find. So this assay looks at dissociation of naloxone from the mu opiate receptor binding pocket. And you can see in the absence of this negative modulator, naloxone dissociates very rapidly. But in the presence of the negative modulator, it basically doesn't dissociate at all for the 20 minutes of this particular assay. So we wanted to see how that might translate as a therapeutic in mice. So this assay looks at the effect of naloxone on morphine. So morphine itself is shown in this top trace, and morphine plus a subtherapeutic dose of this RNAM is shown in green. However, if we have a subtherapeutic dose of morphine, a subtherapeutic dose of naloxone, and we add increasing amounts of our negative modulator, shown here in cyan and purple and dark purple, it's quite effective at having a stabilizing effect of naloxone. So this is our negative modulator, and it could potentially be a therapeutic together with naloxone for opiate overdose. And the last one I want to show you is our positive allosteric modulator. So again, this is the receptor, the structure of the mu opiate receptor. Metenkephalin is a native peptide, and this AP58 binds on top of intracellular loop 2 in this relatively shallow pocket. And I was asked earlier today, would AI basically allow us to develop models that would detect where these two bind? Unfortunately, AI was not able to predict where this pocket is quite shallow, and it simply couldn't detect the binding pocket for this compound. So these are cryo-wave structures. So the first assay what I want to show is a tail flick assay, and it's a very simple assay. It simply uses a light beam to heat up the tail, and if the mouse is uncomfortable, it will start wiggling its tail around. And if it has a level of analgesia, the tail it basically won't move the tail. So here is an example of giving the mouse a subtherapeutic dose of morphine shown in cyan here, a dose of AP58 by itself, the PAM shown here. However, if you take a subtherapeutic dose of morphine and add increasing amounts of the PAM, you now have analgesia. So we can see this works in vivo. And it shifts the dose response curve for analgesia to the left. So a lower dose of morphine is required in the presence of this positive allosteric modulator. And then finally, we wanted to know whether the modulator could work by itself with an endogenous opiate. And for this, we use a chronic constriction model of pain. So the cyanic nerve is constricted, and this generates a painful hind cough. And you can test by poking it with fibers of different strength, the susceptibility to pain. So, for example, the black trace here just shows the susceptibility, high sensitivity to pain in the absence of any analgesia. But simply by giving the mouse increasing amounts of AP58, in the absence of any other exogenous opiate, you get relief of pain. And the relief of pain from AP58 is basically as effective as a known gabapentin drug used for the same purpose. And also, in a measure of addiction, this PAM doesn't cause what we call condition-placed preference. So a mouse is put into a cage with two chambers, and in one chamber it's given, say, morphine, in the other chamber it's given saline, and when you put the mouse back in after a week of this treatment, it will always go to the chamber where it received morphine, and that doesn't happen with AP58. So we think this is probably the most exciting compound that we have so far, and it suggests the real potential for allosteric modulators as being therapeutic options for pain relief. So again, our challenges are, can we develop HAMs that only recognize endogenous opiates? AP58 will also recognize morphine and fentanyl, so it could simply augment the abuse of those drugs. And then can we develop more pathway-selective PAMs that will probably be safe? So I'll end here and just mention that I had, as I mentioned earlier on, a great series of collaborators in my lab at Stanford, and particularly Bill Weiss, who unfortunately passed away 2 years ago, was a fantastic crystallographer and a great friend, and someone who really helped me get engaged and do the right experiments to help us get crystals of the beta-2 receptor. So I'd be happy to take any questions.
For that inspiring and insightful lecture. Please kindly return to your seat. We will now have a 10 minutes break before the Q&A session begins. The Q&A session will be conducted via Slido. Please scan the QR code to submit your questions and upvote those you find most interesting. 本场演讲的 Q&A 提问将使用 Slido 平台进行。请各位与会者扫描现场提供的 QR code， 即可进入提问页面，提出您的问题或对其他问题进行按赞。Welcome back. We would now like to invite Professor Kobilka and Vice President Professor Liu to the stage for an interactive Q&A session. We have also prepared a selection of questions submitted in advance by participants during online registration.
Come back, and so you can use the Slido and to ask the question. So before we started, I take the privilege to ask several questions. As we know, you say you screen like almost billion, so that's the in silico screening.
So yeah, in silico screening is now roughly $1 billion.
Maybe. Okay, here. Maybe use this.
So the in silico library is now roughly 1 billion compounds. What I didn't mention is that that's a very large number of compounds to handle. So for example, the scoring function on in silico screening is not so good that it can, let's say you have to pick the top 0.1% of the compounds will have a very similar scoring. And so let's say you screen a billion compound library, you might have to go through a million compounds to find something that actually works. However, Brian Schreckett has worked a lot of this out and he knows ways of paring down that million high-scoring compounds. But it's not something that's readily, for example, my lab doesn't have the expertise to do a in silico screen because of those issues. However, a billion, we screened a 30 billion compound DNA encoded chemical library, and we only had to pick 10 compounds, and we got several hits. So the scoring function for in silico for DEL screening is much better than for in silico screening.
Okay. And one more question is about. I think we all spend a lot of time to learn how to get crystal, how to clone the protein, and do the X-ray crystallography. But this day, people use the AlphaFold, the AI, to get I don't say the structure, to get a model, right? So how do we tell our students to spend a lot of time to study the biophysics, but you can get a structure just a few buttons away from the AlphaFold?
Well, as I mentioned, AlphaFold doesn't work for everything. For our PAM structure that I showed at the end, AlphaFold couldn't predict where that molecule was binding. So it's particularly good for inactive states, for antagonists. But there are other things that we're interested in. So, for example, I mentioned there are these intermediate states. And we have physical evidence using single molecule fluorescence spectroscopy that we can actually see these intermediate states going back and forth. The problem is that the lifetime of the intermediate states is at most 1 second. And what we're trying to do is find conditions where we can lengthen that residence time for these intermediate states. And in doing so, we could then go to cryo-EM, and it will, it might be a small fraction of the particles, but we'll likely be able to see what the structure of this intermediate state is. You can't do that with AlphaFold.
Okay, let's take the first question. Okay, over the course of a long research career, how do you sustain this enthusiasm and the motivation for your work?
Yeah, I get asked this a lot. Like, why did it take you 17 years? There's sort of a process of failure. I mean, you try something, you fail, and if you don't just fail, but you try to figure out why you failed, you learn something, and then you can move on to the next step and the next step. Obviously, over 17 years, there are many failures, but I learned a lot about the protein, and we weren't simply just trying and failing. Once we could make enough protein, we started doing biophysical experiments and learned something about how these receptors work and why it was so difficult to get them to crystallize. We learned that they were just highly dynamic, and that helped us in a number of ways. It helped us to find drugs that reduce this flexibility and also design protein engineering methods that reduce flexibility, and that ultimately led to success in getting structures. But it was just exciting to learn anything about these molecules. So even the biophysical experiments, which I really still love, and we're doing more of them now even than we did in the past, using single molecule spectroscopy. But there was just always an excitement of making an incremental finding about the behavior of this protein.
Okay. Another second question. So in your view, there are limitation of AlphaFold or AI in capturing the dynamic multi-conformational behavior of GPCRs, particularly in relation to bias agonism. Can you answer some of that?
Yeah. So, you know, I don't know, you might be successful at some point. So for example, we haven't compared AlphaFold potentially giving intermediate states. It often gives you a number of different structures and gives you scoring functions, so you know which structure has the highest likelihood of being correct. But we've had enough, I don't want to say failures, but inability of AlphaFold to predict particular structures. Particularly, it's in our hands, it's been less effective at predicting active state structures. And those intermediate states are very unstable. So I think by just the nature of their conformational complexity and dynamics behavior, they're not good candidates for AlphaFold yet. I'm not saying they won't be. But whenever we have a new agonist, we just simply go to cryo-EM. Right now, if we have cryo-EM time, it's so fast to get structures.
So how do you measure the balance between fundamental academic research and drug development? Maybe in your research work in the company?
Right, so as I showed, we're doing some drug development in the lab, but we have a small biotech company that's run by my wife, Tamkang, and really, talented people. I don't need to spend much time at all with conformetrics. I'm available. We have a meeting every week. I follow what's going on, but the scientists are so good. They've been at the company for such a long time. And Tungsan takes care of all of the administrative work as well as some of the scientific issues. So it's not a big time commitment for me.
Okay, I was learned from the Nobel Laureate website, and you told them like you learn how to manage the lab from your father, and see how he managed his bakery shop.
Okay, yeah. So my dad owned and operated sort of a moderate-sized bakery in a small town, and it made an amazing collection of pastries and cookies and cakes and breads, and he managed to keep this high quality of baked goods in part by being a very good boss. And when I say this, he was very fair to his employees, but he also knew what everybody did, and he had to be able to do what everybody did. So if someone either didn't show up or quit, or my dad would fill in until he found somebody to take over. So I think also the way I work with people, and also particularly early on in my career, I was pretty much able to do whatever anybody else in the lab did, and I was very active at the bench for the first 10 years and moderately active for the next 12 years. Since the Nobel, I've tried to do a few things to keep, do some simple experiments, and I love being at the lab, but I'm no longer quite as competent as I used to be. But and so those features that my father, just being good at engaging everybody, knowing their strengths and weaknesses, and being able to do what anybody did if needed. I also really admire my father for one relationship he had. So there was a person who drove a truck to deliver bakery goods, and he wasn't the smartest person, and he would make many mistakes, not intentionally, but it was just, it was sort of a taxing job for him. And my father would not fire him. He simply continued to try to work with him, to try to get him to perform better. And we all, the person's name was Roger, so we all loved Roger. but we also knew he was a great source of frustration for my father, but he wouldn't fire him. Unfortunately, when my father sold the bakery, but within a month, and Roger was supposed to stay on within a month, he was fired. So that was just an example of sort of his compassion, his willingness to work with people, his knowing people's strengths and weaknesses. And I hope that I brought some of that with me when I started my lab.
So in other words, you never kick out your students, you always give them one more chance?
So I have finally got rid of one student, but that was after like 10 tries, 10 chances. I mean, yeah, that was very difficult.
Okay. So what motivated you to start your own company?
I guess, so we started the company before we were able to get structures. But the idea was that, can we use, we by that time we had a number of biophysical approaches to looking at conformational changes and structure dynamics. And we thought, well, is there some way of applying this to drug discovery? So we started the company. It was entirely, we didn't have any venture capital money. We didn't take any money from relatives. It was, we started out and got some contracts from various pharmaceutical companies, and for the most part, up until recently, we were relatively well funded from being sort of a CRO, contract research organization for structure of fairly big pharmaceutical companies. But with the sort of advent of the availability of Dell libraries, we started doing drug discovery ourselves. And so now we are a small but somewhat successful small molecule drug discovery company.
Okay, so when you founded Conformate RX, what were the most critical forms of support from Stanford? Unfortunately, our president is not here, otherwise he can learn from that. In the early stage of fundraising, how can founders effectively attract the attention of an angel investor? I think we have some investor here.
Oh, okay. We've never, we really haven't tried. We certainly haven't had support from Stanford, but we didn't go out fundraising. As I said, except for small business grants, we've been very successful in getting small business grants, and that got us started and kept us going until we started getting contracts with pharmaceutical companies. But we're quite unusual in that even up until today, we don't have any venture money, but we hope to change that soon.
Okay, next. You have the next question. So, are there currently any open postdoctoral position in your lab, and what qualities do you look for in applicant?
So I get about 2 or 3 applications a day. So what I look for in the postdocs I currently have in my lab and in the past, typically they write an email saying they love my work. But what's most important is they say, this is what I would like to do if I could come to your lab. And it shows that they've gone just beyond, stroking my ego and saying I'm great, but really what they feel are questions that remain unanswered that they'd like to answer. And almost all of the postdocs who are in my lab have written that kind of email. Now, one could say, okay, I get an email from a postdoc and I said, well, you can write to me and tell me what you want to do. But I'd rather have them just do it. And at least that has been fairly successful in finding postdocs who are both talented. And obviously they have to have some record of achievement in their graduate work.
Okay, can you slide over? Or do we have more question? Hold on a second. Give him a microphone. It's my tennis partner, so I need to. He's a professor in chemistry department.
Okay, I'm Professor Ruben Hu. We have a common experience at Stanford. Okay, I graduated in 1982. You went there in 1988, right?
At the very end of 1990.
Okay. You mentioned the entrepreneur's experience. Actually, Stanford is very popular for that. So many professors there has their own companies. Especially, the neighbor is Palo Alto. Okay, that environment is good for it. So many people will benefit for it. So how do you think your chance when you start your company at that area?
So at the time that we started our company, there was the feeling, at least among many of us, that we had to really separate company from the lab. And so that's more or less what we did. But I wouldn't say Stanford discouraged me from doing it, but I had to make sure that anything done in my lab at Stanford did not benefit the company, other than just expertise. So we couldn't work on the same proteins, for example.
Okay, thank you very much.
So the next question. So in the new era of drug development, how important is sustainability, and how can it be practically implemented in the R&D process? I think it's sustainability.
Yeah,
Like
I'm not quite sure I understand.
SDG 17 or something.
Yeah, I'm not sure quite how important is sustainability. Sustainability, I'm sorry, I don't really.
Environmental protection in terms of the whole human being, cultural sustainability.
Oh, okay. To be honest with you, I'm not such an expert on drug discovery that I can really properly address that, I'm afraid. We're very early in drug discovery efforts in my lab at Stanford, and even at Confirmetrics we're further along, but I wouldn't say we have a lot of experience with drug discovery, particularly beyond the identification of novel compounds.
The next one, which assumption about GPCR activation turned out most misleading in your work?
So assumption, I'm not sure that I ever really made assumptions about GPCR activation. Most of what we know or what I've assumed comes from experiment. But I think maybe this is too focused, but we've thought all along that there's a general mechanism for activation. All GPCRs, when activated by agonists and partial agonists, will lead to an opening of transmembrane 6. And recently we found that for the opiate receptor it's very different. It has, with the exception of these very potent, lethal opiates, the opiate receptor has some other mechanism. Because TM6 does not move outward upon activation, only when G protein engages. So that's probably maybe a misleading assumption about activation.
Okay. How does the AP58 partition into the plasma membrane from the solution? How specific is the AP58 to receptor?
Those are really good questions. So AP58 is a really interesting compound. I could have spent 10 or 15 minutes on it. It started out as being a positive modulator from a screen from Hsing. You're all familiar with Hsing as well? Hsing, it makes fantastic DNA-encoded libraries. And it was a very weak PAM, And we sent it to Sushoda Majumdar, our medicinal chemist, and he made, I think he's made like 50 compounds, 50. It has a scaffold that's very amenable to a modification. And so he finally got AP58 to become a very efficacious PAM. The problem is that we could, these are all in biochemical assays. So because we use a purified receptor to screen the library, it's possible that our modulators would bind on the outside or bind on the inside. And as you point out, this binds on the inside, so it must have to cross the plasma membrane. And we tried a number of different cell-based assays, and we could not see an effect of AP58 in cells. So Sushoda Majumdar just sort of ignored that and went into animals. So he went into mice. And we immediately saw that it works. The data that I showed you is efficacious both in enhancing the potency or the efficacy of subtherapeutic morphine and even endogenous opiates. So this is still sort of confusing us. It must get across the plasma membrane. It does. It has to have an effect in mice. We are now seeing some effect in cell-based assays. One of the problems is that we've started relying so heavily on modified G proteins in various signaling assays that I think if you have a relatively weak, efficacious compound, you really need wild-type signaling proteins. So if we use the wild-type GI as opposed to bread-based assays and look at inhibition of cyclic AMP, we can now see some effect in this. So it must cross the plasma membrane. It's lipophilic enough to do that.
So it's looking very promising, but I mean the A58. And so if everything is fine and the clinical trial, then how long you think to take me in the market?
I mean, you heard the numbers earlier today. 10 years is not too long. I should mention I didn't completely answer AP58. So AP58 is not entirely specific for the mu. So we have to make further modifications. So the pocket itself, there's some differences between mu, delta, and kappa. And we believe that it would be possible to further modify it, but we might need a different compound. But the other interesting thing about this is it doesn't have the effect on agonist binding as you would think. But we were learning from other allosteric compounds is that they sometimes have dual properties. So there's an allosteric compound for the cannabinoid receptor that enhances agonist binding but inhibits G protein coupling. So we're starting to see just sort of weird behavior as a result of this completely new class of compounds. But I'm not sure AP58 is going to make it to the clinic, but but the fact that we identified it and it has such interesting properties compels us to do more screening.
Okay. So which is the happiest? Cycling with Jason, forming a family, discover GPCR, or one Nobel? I want to know what really matters in life, in your opinion. I know you do the cycling in Tour de France with Jason, right?
Yeah, I would say that's my highlight. Interestingly, it came after I was at the synchrotron in Grenoble, and it was Tungsten and I were both at the synchrotron, and it was where we saw our first diffraction spots. So these are our very first crystals were obtained in 2005, and they were ugly, and they didn't diffract very well. But we would take turns putting crystals on the beamline, and I would be sleeping, Tungsten would be doing work, somebody else would take over, and Tungsten saw the first diffraction spots. So they were only like 20 angstroms but they were clearly protein crystals. And with that data, I actually was able to finally get a grant to support my crystallography efforts. But, and then after that, I met Jason in southern France, and we rode the Pyrenees stages and watched the tour, and that was amazing.
Okay, get a crystal. I think the membrane protein is not easy to get a crystal. I say when you just mentioned when you get the crystal, and most of the time we get the crystal like sodium chloride, not the real crystal. I think Professor Julie know when you get the first membrane protein. I think the joy must be grateful. Okay, thank you. The next one. Do we have a next one? Did you ever consider giving up on your experiments because of poor result? If so, how did you handle it?
I don't think I ever thought of giving up. And as I mentioned, I was learning things along the way. And we were able to publish some work on our biophysical studies. And it was just it was a challenge. I mean, it was every failure. So what I found was really important, when you design an experiment, assume it's going to fail, and try to include in that experiment ways of troubleshooting. So if it fails, you can at least learn something. And I learned a lot from those failures. So I never thought of giving up. It was always great having a lab of young people working together with me on a number of different projects. So although it was my favorite project, we were doing other things and contributing to the field.
Okay. And how many time we left? 15 minutes. Okay, we still have some more questions. Next. Scientific breakthroughs require a unique level of critical thinking. What specific habit did you cultivate early on to train your mind?
I don't know. I didn't do it. I didn't intentionally try to train my mind. But I do think that the fact that I love to be at the bench and do experiments meant that I wasn't helpless. I could do things that I wouldn't ask a postdoc or graduate student to do, because sometimes I'd be embarrassed to let them know what I was doing because it was so unlikely to succeed. But yeah, I definitely didn't train my mind.
Do you think we can use a proximity concept like PROTAC, P-R-O-T-A-C, to force GPCR and the specific G protein subtype together to bypass off-target effect? I'm not sure.
Sounds sort of like convoluted. I mean, you might be able to do that in a cell-based assay, but you're not going to be able to do that as a therapeutic. So I hadn't thought of it. It's an interesting idea.
Okay. So next. Do we have next question? What could be the most frustrating event in your career, and how did you get through?
I think the most frustrating part of my career was I was fortunate to have Howard Hughes funding when I started my lab, and I don't think they thought I had a chance of succeeding in getting a structure. So in 2003, they terminated my funding. At the same time, I could not get a grant to support my structural work. So I thought we were making progress, and I didn't have enough money, and I got deeply in debt to the university because I overspent on my grants. So that was probably the most frustrating time for me in terms of the research. Fortunately, Stanford could have said, look, you got to stop. You can't spend any more money. They didn't. They gave me a loan, and then we had a number of gifts from people who I knew in the pharmaceutical industry, and we were able to get through that difficult time. But it was right after that that we got our first crappy crystals. But you know, and that really, seeing those first crystals diffract made me think, well, we just have to go back and optimize those conditions and we'll get a structure. They never did diffract any better, but at least it told me that you can make these things diffract.
What is the advantage of PAM or NAM over also sterics ligand?
So I'm not sure, I can't really think of a great clinical application for a NAM other than, you know, maybe it could reduce the body's response to, say, fentanyl. And if it was a very long-acting compound, it might be a way of avoiding abuse. For PAMs, though, I think there are a number of advantages. Particularly, let's say the PAM only works with metencaphalin or endogenous opiates. What it would do is, I've broken my leg, I have pain, and the PAM is augmenting when I feel pain. When I'm at rest and I'm not particularly feeling pain, they won't act by themselves. So they have a temporal action and also the effect on enhancing responses to endogenous opiates. So that's why we think they're potentially valuable therapeutics. As opposed to orthosteroid compounds, which are, you take a pill, and as long as you're above the half-life, you've got drug in your system, even if you're not feeling pain anymore. So I think they're less likely to be addicted.
Protein structures are often soft in a bound state. Not really. This may limit the evaluation. What do you think the limitations of a current structure-based drug design are? Not really a bound state. I think you solve a lot of the free state.
Actually, it's very hard to get the free state for most receptors. But there have been some that have been solved in the free state. I can't tell you exactly which one it is, but there's one that's been solved in the free and the bound state. In the free state, you wouldn't predict where the binding pocket is, so one of the extracellular loops folds into the binding pocket, so that free state structure wouldn't help you with drug discovery. I think the potential problems with, let's say you're bound to a full agonist, and what you really want is a partial agonist, and you're using the structure of the full agonist bound receptor. Maybe that's going to bias you in your screen towards getting another full agonist as opposed to a partial agonist. But I don't really see any necessarily severe limitations if you know that you're going to have to optimize whatever compound you get out of a screen.
Okay, the next one. So how was it determined that fentanyl can be used to treat elephant? Are the GPCR in elephant being studied?
I don't. The answer is, so fentanyl isn't used. It's low fentanyl. So low fentanyl is about 100-fold more potent and even slightly more efficacious than fentanyl. Surprisingly, we started using it a number of years ago when it wasn't a controlled substance. So we were able to purchase it, and now it's highly controlled. But it's so dangerous that people in my lab can't use it unless somebody else is in the lab, and we have naloxone in the lab. We don't use it that much anymore. But I think if you're going to anesthetize an elephant, you need a huge amount of fentanyl and maybe 1/10 of the amount of low fentanyl.
Okay, next question. Can you describe how Nobel Prize changed your life? What advice you think junior research should do in present day? I don't know what you mean present day, maybe short of a grant, short of a student. For juniors.
So I don't think it should be anybody's goal to win a Nobel Prize. There are so many incredible scientists who have made major contributions who, just for want of enough Nobel Prizes, won't get one. So what advice? I would, what's really important is to find something that you're really interested in and a challenging problem to solve. If you can't go into work every day, hoping that you'll make some progress, it'll be hard to sustain yourself. So, I'm not even sure I thought of Nobel Prize. If you asked me when I joined the Lefkowitz lab, what are Nobel Prizes and how many of them are there, I wouldn't have known. So I had no thought of it. But I did find something I really wanted to pursue, and I hope that everybody can do that somehow.
Looking back, were you driven more by curiosity or by a sense of impact? And what habits or ways of thinking matter most beyond talent?
So it was definitely curiosity. Yeah, I guess curiosity and the desire to make some incremental improvement. I knew that fairly soon after, it took me 5 years to be able to purify enough protein. I knew this wasn't going to be a quick fix, that it would probably be many more challenges that I'd have to overcome. But as I mentioned earlier, I loved the taking on the next challenge and making some incremental advance.
Okay. The next question. Okay. Finally. Let's. I know you are the member of a BioX program in Stanford, right? And I think the BioX, we heard that programs. I think before 2000, it's almost the first interdisciplinary program, and I think a lot of universities, including Tsing Hua, they start to adapt similar program, like to promote cross-discipline collaboration. But I don't think any universities get success like Stanford. Can you comment on that?
I'm not sure how much BioX has contributed to sort of the interactions between various faculty members. Stanford is somewhat unusual in that it's a relatively small campus. The medical school campus and research facilities are right across the street from biology and chemistry, and so you don't have to walk very far to find a collaborator. And for a number of reasons, it's always been a highly collaborative environment. And I've never had trouble finding someone who helped me out or gave me advice, or as I mentioned, Bill Weiss early on would just, I felt like I was one of his graduate students. IOX sort of reflected, I think, the state of what existed already, but also made some funding available to further cement interactions between individuals. But Stanford's an amazing place, and I was really fortunate to get a job there.
Okay, thank you for your interesting and inspiring talk, and also the very interesting question.